Today we are recognizing Dr. Jeffrey Gordon with the Cobra Medal from the Association of American Physicians. And I am the very lucky colleague and friend that gets to introduce him. He is the Dr. Robert J. Glaser Distinguished University Professor and Director of the Edison Family Center for Genome Sciences and Systems Biology at Washington University in St. Louis. I believe we are recognizing Dr. Gordon for revolutionizing our ability to study the gut microbiome. His work on the microbiome has transformed our understanding of gut function in a fundamental way and has forever changed the field of nutritional sciences by showing that it is possible to design foods that promote health on the basis of an individual's microbiome. His discoveries have profound implications for the health of the public at a global level. Like any of the important sustained scientific revolutions in our history, his work is built on the essential criteria of the scientific method, including the bold use of new technologies to challenge the null hypothesis and novel analytical approaches to provide unbiased quantitative proof. I find it truly amazing to think back now to 20 years ago when sequencing the human genome was a revolutionary accomplishment, and each of us would talk about how much harder it was going to be to sequence our microbiome. Fast forward to present, in which, thanks to work by the Gordon Lab and others around the world, we are not only capable of sequencing bacterial, viral, and fungal genomes in the human ecosystem, but also able to analyze in an intelligent way that massive data that comes from it, to carry out the most sophisticated profiling of gene expression, and to be able to pick up important metabolic signals from the most obscure microbial constituents amongst billions of organisms that inhabit ever tinier niches in our gastrointestinal system. Looking back on it now, it took enormous guts for Jeff to take on the path and journey of the microbiome. It is especially fulfilling to be here today because his contributions validate so much of our professional identity as physician scientists in the fullest iteration of that identity. And it feels so right to be honoring his accomplishments in the public health area of nutritional sciences at a moment in history when public health has been once again rightfully recognized for its utmost importance to the functioning of our society. Jeffrey Ivan Gordon was born in New Orleans on October 4th, 1947. Early on, he was fascinated by microbes and space travel. He attended Oberlin College and then University of Chicago for his MD. He then went on to residency in internal medicine and fellowship in gastroenterology at Barnes Hospital and Washington University. Like so many of his generation who wanted to pursue careers as physician scientists, he spent 3 years at the NIH as what Bob Lefkowitz will describe later today as a yellow beret. He returned to WashU, where he has remained for his entire career. By the way, Jeff keeps a framed copy of his letter of rejection from WashU Medical School to emphasize to his students how career paths can take unexpected twists and delightful surprises. From very early on in his training, Jeff's special capabilities were noticed and nurtured by the most famous and influential academic leaders of the time, including Dave Kipnis, Louis Glaser, Ollie Lowry, and Phil Needleman, and they became his role models and treasured mentors. In 1981, as an assistant professor, he started his own lab in the Department of Biochemistry. As has been his modus operandi throughout his career, he began what I call the three-pronged approach of Gordon. First, a daunting goal. Second, courage to learn and apply the most powerful new technology. Third, engage the most talented, of the WashU, MSTP, and PhD students. The goal in this initial period, which spanned the years 1981 to 1997, was to better understand how the cells of the intestinal epithelium develop and differentiate, and what determines how they function. He used transgenic mice in a completely novel way. This work generated new insights about intestinal function under homeostatic conditions, but also through unique models, led to revelations about inflammatory bowel disease and neoplastic disorders of the intestine, colon, and stomach. It was not only pioneering, but has led to long-standing and foundational shifts in the fields of gastrointestinal physiology, developmental biology, and pathobiology. Sometime in the mid-1990s, Jeff began to launch work on the microbiome and how that could be incorporated into understanding gut function in health and disease. The big step here was the nodobiotic mouse model and carefully controlled introduction of bacterial strains and dietary constituents in a way that would permit both a reductionist approach with rigorous quantitative analysis, but also to provide system-level understanding. When this was combined with the newest functional genomic analytical tools, a historically unique experimental system was established. In a landmark paper published in Science in 2001 with then postdoc Laura Hooper, he showed that the system could be used to characterize how commensal bacteria inhabit and play a role in the essential functions of the intestine. Over the next several years, results from these microbiome explorations led Jeff to propose in a 2005 white paper to the NHGRI, a human microbiome initiative that would deliver genome sequences for 100 gut microbiota members, herald another phase of the Human Genome Sequencing Project, and provide a key reference for subsequent microbiome projects. His white paper and advocacy laid the groundwork for NIH's Human Microbiome Project. Later, the Gordon Lab learned how to transplant human fecal flora samples into the nodobiotic mice and utilize samples from twins to address potential confounding variables of genetic versus environmental factors. In 2013, this work culminated in a study published in Science using fecal samples from carefully characterized human twins, showing that the metabolic effects of obesity could be conferred on a mouse by the intestinal bacteria of obese humans in a diet-dependent manner and could be reversed by the intestinal bacteria of lean individuals. These results illustrated a means for establishing preclinical proof that microbiota changes are causes rather than effects of altered physiology or disease, for identifying microbial therapeutic targets, for developing microbiota-directed foods, and the concept that such foods could help address the intractable public health problem of obesity. During the era of 2000 to 2010, Jeff and his colleagues also began to focus on what happens to the gut and its microbiome in normal development, and then what happens in the vicious cycle that accompanies infantile diarrheal diseases in impoverished communities all over the world, a cycle that impairs growth and development and increases susceptibility to many adult disorders. They established approaches for transfer of fecal samples from affected infants that could reproduce in the nodobiotic model healthy development, but also growth stunting from diarrheal diseases and malnutrition. 3 major advances have taken the work to new heights. First, from careful attention to the microbiome in both healthy development and the dysfunctional state, Gordon and colleagues showed that a gene expression pattern of 15 very rare bacterial strains could be used to benchmark the health and maturity of the gut microbiome. Second, his team discovered that the gnotobiotic mouse model could be used as a preclinical model to design foods that are complementary to and could sustain these healthy bacterial strains. Third, the Gordon team established a clinical trial platform in collaboration with Tamid Ahmed of the International Center for Diarrheal Disease Research in Bangladesh that allowed carefully controlled clinical trials of what are now called microbiome-derived complementary foods in children with severe acute diarrhea and malnutrition. Together, these developments made possible the results published over the last 5 years in Science, Cell, and the most recent 2 studies in New England Journal of Medicine showing that unique foods designed and manufactured based on their effectiveness in the preclinical model outperform World Health Organization formulas in resolving diarrhea and growth stunting in malnourished children. These landmark studies establish and usher in a new era in which foods can be designed in a personal way to improve health. It is relatively easy to see how these findings might impact the underdeveloped world, but I would argue it is an overall strategy for improving health in the developed world as well, now possible to envision for the first time. I want to repeat this conclusion out loud again because it is so important. Foods can be designed in a personal way to prevent impairment of infant growth and development and to promote health. This simple phrase is the legacy of Jeff Gordon's accomplishments and represent the ultimate in going from a twinkle in the early phases of basic science to clinical proof of concept and efficacy using the bests of the personalized medicine paradigm. I want to say a few more words about 2 traits that distinguish Jeff Gordon and deserve emphasis. His devout commitment to the scientific method has led to incredibly powerful tools, including extraordinary model systems and computational approaches that can be combined with genomics, proteomics, metabolomics, lipidomics, and analyzed after precise experimental manipulations. This commitment lays the groundwork for investigators and labs around the world to draw scientifically sound conclusions and to advance the field for years to come. We have already come to know that the microbiome plays a role in human health and disease that is much more broad and deep than anyone predicted or even imagined. And Gordon's foundational work has allowed us to realize this without any hype. Second is his dedication to mentoring and education. I first came to know Dr. Gordon when I moved from Children's Hospital Boston to WashU some 36 years ago. I revered him, and several years later, when I became Chief of Gastroenterology and Nutrition in the Department of Pediatrics, I was asked for a vision statement. It didn't take me more than a second to prepare a single sentence, build the division with MSTV graduates from the Gordon Lab. Jeff has attracted and mentored well over 130 students and postdocs, many of whom have gone on to renowned careers in microbiome research as well as other important areas. There are too many to mention here, but suffice it to say that perusing the first authors of his more than 500 publications is like a walk through a list of superstars in biomedical research. Jeff's work has been recognized by many organizations. He is a member of the National Academy of Sciences and the National Academy of Medicine. Of his many awards, I know he is most proud of the Copley Medal of the Royal Society of London, the Sanofi Institute Pasteur International Award for Biomedical Research, the Rhodes Medal for Distinguished Achievement in Medicine of the American Philosophical Society, and many others. I want to end my remarks and introduce Jeff by speaking a bit more poetically. In our culture, gut is one of those words that can refer to two things we usually think of as completely opposite. In the earliest meaning of the word, guts referred to bowels or entrails, the physical viscera of the human body. But the word gut has also come to be synonymous with what is more abstractly visceral. We use it to refer to a person's truest, most basic feelings. Quote, check in with your gut, we say. We ask, what is your gut instinct? Of course, we all know that what so many refer to as gut feeling actually comes from our brains, and when we use the word gut in that way, we associate it with what is most instinctive or emotional, rather than that which is rational and completely thought through. In many ways, the opposite, in terms of connotation of what we do as scientists, what Jeff does as a scientist. I'm telling you all of this because it is useful in trying to adequately describe just how astounding Jeff's body of work, if you'll excuse the pun, really is. What Jeff has done is nothing short of revolutionary, in part because of his dedication to combining those two approaches in his study of the literal gut. He allows his team to be guided by their instincts and imaginations, even as they investigate their hypotheses using the most rigorous and cutting-edge tools science has to offer. In doing so, he has become not just the father of the microbiome, but a mentor to so many scientists who understand that delicate balance between the concrete and the theoretical, the parameters of the world in which we live, and the potential of science to expand or shift those parameters, leading sometimes to a completely new understanding of that world. I think Jeff himself sums this idea up quite beautifully in an essay he wrote about his journey in science. And I quote, "In looking back to recount the lab's journey, I feel today what I have always felt, that I have been incredibly fortunate to have been surrounded by an inspiring group of remarkable students with enormous curiosity, a deep commitment to learning, the courage to explore the unknown, and a willingness to cross traditional disciplinary boundaries to obtain answers to questions they found to be captivating. They have had a wonderful sense of community, a shared belief that discoveries and innovation are born from a caring, supporting, respectful, and trusting environment where we can communicate our ideas freely with one another and not be afraid to say, I don't understand, so that we learn together. They have manifested that wonderful mix of idealism, imagination, and other directedness that allows people to look beyond themselves to embrace and together pursue with hope, modesty, and humility, using both basic and applied science, solutions to challenging global health problems that are necessary to approach now rather than later and for sustained periods of time." End quote. I now would like to introduce the 2021 George M. Kober Medal recipient, my friend and idol, Dr. Jeffrey Gordon.
Thank you for this wonderful award, which, given its extraordinary history of recipients, I accept with the deepest appreciation, humility, and gratitude on behalf of the magnificent group of students, postdocs, and colleagues I've been so fortunate to have worked with and learned from over the years. When I was young, I met my wife, Deborah. She was a freshman at Oberlin College, and I was a junior. She and our two children have transformed my life in more ways than I can possibly describe. I'm so grateful and so fortunate. When I was a student at Oberlin, I experienced a place deeply committed to a liberal arts education. I experienced people who looked beyond self and were dedicated to understanding the foundations of human well-being, of what features in society enable people to flourish, I was surrounded by individuals who were dedicated to understanding and resolving problems related to social justice, to understanding how we can care for one another and live together peacefully. When I was a young faculty member at Washington University in St. Louis, I encountered extraordinary mentors, notably David Kipnis, a winner of the Kober Medal and former head of the Department of Medicine. David had the ability to see the world through another person's eyes and to place young people in environments that were most supportive of their needs and strengths. David, like all great mentors, had the ability to empower people by conveying a sense that they were valued for who they were and what they stood for, not just for the work they produced. I'm so grateful for the kindness, thoughtfulness, and wisdom of my mentors. Their lessons have never been forgotten. When I first joined the faculty, an MD PhD student knocked on the door of my newly opened lab and asked to come in. He was my first graduate student. Looking back, I'm so grateful to the remarkable group of 138 PhD and MD PhD students and postdoctoral fellows who've come to the lab to share several years of their lives with me. They have had beautiful dreams and ambitious goals. This century presents great challenges for humanity. We will learn whether we have evolved socially, psychologically, and biologically in ways that are necessary for us to be able to care for one another. Seeing global health challenges embraced by students in ways that combine hope, humility, passion, empathy, courage, creativity, and a commitment to innovation gives me hope for the future. Current human microbiome research is addressing questions originally posed by microbiologists more than a century ago, but it is doing so with new and rapidly expanding sets of tools. We are witnessing its inherently interdisciplinary nature, which provides an opportunity to forge new alliances, spawn new fields, and transform the ways we educate ourselves. We are seeing how this research can inspire students to expand their understanding of the human condition. The public is captivated. As such, there is a need for those in this field to describe their findings clearly, in a sober fashion, to approach bench-to-bedside translation carefully, mindfully, and to engage in a proactive societal dialogue about the ethical, legal, social, safety, and regulatory issues raised by this research. I feel young, although I'm older now. It is because I believe that all that has transpired, all the work that we have done, makes it possible to just now begin to do what we have always dreamed of doing, to begin to start understanding. The best part of the journey is now and will be tomorrow. Thank you.